Okay, welcome to Chapter 18 recording on gene influences or genetic programming that happens between the point of DNA turning into a protein. So what we're mostly focusing on here, just so you kind of get a gist of this, is that we are going to be focusing on the central dogma, which is basically DNA turning into RNA and then turning into protein. Now remember that DNA is considered your genotype and protein is considered your phenotype, so how you look. And RNA for the longest time was all about the helper, I guess, but we're finding out that there's a heck of a lot more of an influential role for RNA. So we're going to be talking how can we influence things from this point to this point. So we'll work through different things like operons, which we originally thought were in bacteria, but we're finding in eukaryotic cells, to histone acetylation and methylation of DNA components, to homeobox or homeotic genes and different groupings that we've found that are controller or light switch type of genes, to RNAi or RNA influencing or interference, I should say, not influencing, and this will be a big piece of our big story on how gene control is all done. So first thing right here is an operon. This is a Lac Operon from a bacteria. So you're looking at a section. Here's RNA polymerase. Remember, RNA polymerase is going to be making the RNA, specifically the mRNA is what it's going to be doing. Now keep in mind that bacteria are prokaryotes, so there's going to be no RNA processing after this, so there's no spliceosomes with them. But they do have environmental controls that govern whether or not it will make the enzymes that are found on its DNA. So what you're looking at right here is the beginning or important area, the operating system for making the very beginning here of the first piece of the enzyme lactase. So lactase will work on lactose. So environmental influences, if there's lactose present, then lactose should be made. Now, lactose isn't as good of a sugar source as glucose is. It's a disaccharide. Glucose is a much more beneficial food supply for bacteria. So if glucose levels are high, then molecules called cyclical AMP are not produced. If molecules of glucose are low, then cyclical AMP increases. So cyclical AMP will work with this molecule right here called CAP, and cyclical AMP binds in and makes active CAP. If active CAP, right, cyclical AMP process, will allow for binding into the promoter region. Remember we talked about way back in DNA transcription, translation, there's a portion of our DNA called the Tata box, which is the spot where RNA binds into, RNA polymerase, I should say, binds into. Same thing here. We have a promoter region. In order for RNA polymerase to bind in, as you can see, CAP has to be bound in. So we say that cyclical AMP is an activator for this operon. So let's say that glucose demands, or glucose in the environment for this bacteria is low. That means it produces lots of cyclical AMP. Now, the other thing that goes with this operon is that you end up having pieces like this, which is a repressor, that might bind into the operator. So if this is bound in here, you see RNA polymerase, even though it has this CAP component that's activated, it will not progress forward to make this RNA transcript. So the operator has to be free. What frees the repressor from the operator? It's lactose, environmental pieces. So this piece right here is lactose. We'll put a big L on it. When lactose is in the environment, it takes the repressor off the operator and allows RNA polymerase to move down the line and make the functional mRNA that is going to be used and then finally translated into lactase. So it can break down its lactose sugar, because remember, lactose sugar is a disaccharide, so it needs to cleave the bond, the covalent bond, between the two monosaccharides in order to use the glucose components that are in it. Okay? So this is how operons work. Another example of an operon would be a co-repressor type of operon where the operating system is open all the time, but if environmental influences are around, then it actually turns off, or activates, I should say, the repressor, turning off the operon. So in this example, we have tryptophan that was produced in the environment, and it binds in with the repressor, so we consider it a co-repressor, activates the repressor, and now the repressor can attach to the operator, and now the operator is bound into, so RNA polymerase, that's what this is, RNA polymerase. Oh, this is hard to write with this pen, I apologize. RNA polymerase will be able to try to get in there to the promoting region, but because the operator is bound to, no RNA will be made. All right, let's move on to histone and acetylation, and then the methylation of the DNA. Another way to control whether or not, especially in eukaryotes, if there's going to be transcripts made, if there's going to be mRNA made. So as we know, if this DNA, right, so this is what the black stuff is here, the black line is DNA, and these are the proteins, which we call histones. Remember that histones plus DNA make chromatin, so it'll be wound up very tightly on here. If the DNA is wound up all tight, RNA polymerase cannot get in there and make RNA. So DNA has to be exposed in order for this to happen. So we're saying transcriptionally active chromatin means that we can actually make RNA transcripts. So you can see here, because there's acetyl groups attached to the histone, changes the polarity and allows it to kind of loosen up, and we can actually get into spots to do transcription and translation. If there's no acetyl groups on, as you can see, deacetylation, there's no availability to get in there, so there's no transcription that goes on. So that's the example of histone acetylation. If we're talking about methylation, the addition of methyl groups, unlike acetyl groups, will inactivate sections of DNA, not allowing those sections to be transcribed. So methylation is one way, and acetylation is another way. And the reason that this is so important is we have just found this out, I would say recently, that methylation and acetylation all happen because of your environmental cues. What you do in your daily life. If you decide that you want to eat Big Macs your entire life, you're going to change the way that your genes get transcribed and translated. For a long time, we didn't understand what that was all about, but now we clearly see on the molecular level it has a lot to do with this acetylation and this methylation. This is also why identical twins, even though they have the exact same DNA, their fingerprint even is exactly the same DNA fingerprint, they will end up looking different as they diverge over time, especially with different lifestyles. And that's all because their lifestyles include what they eat, what they do. That plays a part on what parts of the actual DNA are exposed for transcription and translation. So you truly are what you eat. The next thing we look at for gene control is homeotic genes or Hox genes. We'll talk about this a heck of a lot, especially when we start moving into evolution unit. And you'll be reading a book later that talks about with Dr. Neil Shubin. He talks about what's called the sonic hedgehog gene and how that plays a part in the leftness and the rightness of body plans, especially in animals. So what you're looking at here is a fly's chromosome and a mouse's chromosome. So we've got an invertebrate and a vertebrate. We've got an insect and a mammal. And you're looking at the Hox genes here. So we got 1, 2, 3, and 4. And here we have common Hox genes, right? In mice, there's multiple copies. In fruit flies, there's only maybe 1 or 2 of certain types of genes that control body plan. A lot of these Hox genes that we have found are in the PAX group, which we talked about in class. And then another type of homeotic gene that we talked about in class was FOXP2, which is with vocal recognition, or not, I shouldn't say recognition, but vocal development in body plans of animals. As you can see here, I picked 2 pictures, and both of these pictures show mutations in Hox genes. So mutated fruit fly has 2 sets of wings, right? So it has a doubling in its thorax region because, excuse me, the gene for control for that part of the body has been mutated. Same thing here, we ended up having legs growing where the antennae are. That's clearly a mutation. So up here where the head is, the legs are growing. So there's a mutation in body plan. So there's a lot of evidences of this. So we're going to show some stuff specifically in birds and their relativity to extinct dinosaurs, and how those genes are still in their DNA, they're just turned off, and how we could essentially turn on those genes and see how their body plans would change. All right, last piece is called RNAi, right? And RNAi, sorry, is RNA interference. So we're dealing with how we can interfere with the RNA that we made in transcription not getting to translation. So there's a couple pieces that are really important here. We start seeing this conglomeration of all these RNAs that are really, really important. We originally talked about ribozymes and how Thomas Cech figured out a lot of your, or I should say a lot of your, one of your first RNAs that acts like an enzyme with spliceosomes. Well, now we're seeing RNAs in this area that play a huge role in how you end up turning out the way you do. Specifically, we originally found it as a part for fighting off viral infections. So viruses have double-stranded RNA, or short hairpin RNA, or microRNAs. And dicer is this protein complex that when it sees these copies, will chop it up, right? Hence the dicing. It chops it up. And in turn, you get these two pieces. So you get small RNAs, and sometimes we call them small interfering RNA. And these small RNAs, one half is going to be used by the protein complex RISC. So both dicer and RISC play a big part in RNAi. RISC will use one of these small interfering RNA halves as a template to find the transcript that matches it, and it will destroy anything that has that exact same one. So this is a way for your body to shut down transcripts. Now, we talked about this for medicinal purposes. This is going to be revolutionary. The ability to turn off transcripts, even if your DNA has issues, is big. So we don't have to worry about changing every single piece of DNA in your body. We just have to worry about turning off the transcripts, right? Turning off the ability to get from the mRNA to actually the protein. So if we figure out what the protein is that's being made that is bad, all we have to do is make a mirror copy of that, give it to your cell, dicer or chop it up. RISC will use it then as a template to find any transcripts that are being made for bad protein, and it'll destroy all the pieces you sent in, but also any piece that looks like that piece. It's a great way of solving a lot of maladies that humans have been suffering from, things like Huntington's disease and different types of cancers. So look out for this in the future for sure. And you definitely obviously have to know how this works for the test. And then the last piece is, here's another image of how this works, right? So we have the double-stranded RNAs up here, the microRNAs. We have dicer that chops it up. Dicer then gives it to RISC. That's what RISC is right there. It's using it, finding the mRNA pieces and chopping those pieces up so that ribosomes can't actually make the proteins that these bad mRNAs are coding for. All right. And the last piece is then how cells actually talk to each other through signal molecule inducers. We have these proteins that are on the surface of the cell membrane. Molecules secreted by one cell will influence the protein doorways on another cell, which causes this signal transduction pathway involving the nucleus, changing how the nucleus responds, specifically the DNA, to make the proteins for that cell. So this is a way that cells can actually govern how they will develop, right? It also governs how mitosis is done with different types of cells. So this is what's we call this cell talking generically, right? So they don't obviously have vocal cords or whatnot, but they do talk to each other via proteins on the surface and molecules that get exocytotically secreted into intercellular spaces, right? So this concludes the recording for gene control. should go with Chapter 18. Follow the Chapter 21 DNA technology if you need some help with Chapter 20. I wish you the best of luck, and see me if you have any questions.